Oh, good evening, everyone. Today is your session number 4. So I'm your TA Dipanshi, and let's begin with the session. So in this session, we'll be discovering or talking about the week 4 topics, and we'll be practicing few MCQs related to them. We will also look into the problems from the week 3 and week 2 as well, which we previously discussed in our session 3. So in week 4, the topics which we will be going to cover are identifying the human disease genes, functional cloning versus positional cloning, mutational screening. We will also look into the complex diseases, and we will also talk about the human genome project, and the HapMap projects, and why the HapMap project was built, what were the reasons behind this, and in the end, we'll also look into the various omics, and then we'll be binding up with the session. If you feel any doubts, then you can ask or you can comment. So let's proceed. So firstly, we'll be discussing with the topics and then we'll practice the MCQs. So first of all, the week 4 unit revolves around the disease. So in this, disease gene identification is the main thing. So firstly, before targeting a disease, we must know that which gene is involved or associated with that disease. So there are various approaches which we can go for to identify that gene which is involved in the disease. So first approach is the candidate gene approach, and it can be applied to any of the disease, whatever the disease is. And the advantages of using this approach is that it is easier to perform for one or two genes. It requires no mapping and it can directly identify the causative variation or the mutation. And the disadvantage that are associated with this approach is that it relies heavily on the current biological knowledge and success rate is very low. So majorly the approaches that identify the disease gene, they require some kind of prebuilt knowledge. So in this, it is heavily depending on the current biological knowledge. So means if you don't have the knowledge about particular gene, then you can't use this method. Then comes gene mapping by karyotyping. So in the previous unit, we have seen that there are many diseases which are identified by using the karyotyping method, in which we look for the chromosomes, how they appear, and what are the differences between the karyotypes of two individuals, where one is suffering from the disease and another one is healthy. So, in that way, also, we can identify the chromosomes and ultimately the region of the chromosome, and at the end, we will be able to find the gene that might be involved in that disease. So, this method can also be applied to any of the disease. The advantages of this method are it is easier to perform, no familial cases required. So normally when we look for the pedigrees, then we require the familial knowledge. So in this, we do not require familial cases and it can detect large balanced events as well. So the disadvantage of this method is low resolution and it only detects the large chromosomal aberrations. Mutation detection requires the second step. For example, in the karyotyping, if the change is very small, then we won't be able to detect it using the karyotyping. So, due to which we'll be further moving on to the next level, that is the mutation detection. So, it requires the second step. So, this is the disadvantage with this method. Then comes another method, that is genetic mapping by linkage analysis. So, mostly linkage studies were also very much higher in the 1980s. It means it was the study which was majorly used to identify the disease genes. So it is mostly applied for the inherited diseases and it is very easy to perform. But the disadvantage is that it requires large families because it is dealing with the inherited disease. So to study any kind of inherited disease, you require the knowledge of the family. So that's why it requires the knowledge of larger families and it often identifies large loci. And in this also, the mutation detection requires the second step. Now comes another approach, that is genetic mapping by homozygosity mapping. So in this, it is generally applied for the recessive monogenic diseases that are recessive in nature. So it is the advantage is that small families can be used, but the disadvantage of this approach is that it is most useful for consanguineous families. So consanguineous families are ones where the cousins marriages happens. So it often identifies the large loci and mutation detection again requires the second step. So it also means if the mutation or the changes that are affecting the chromosome or the loci, it should be large. Then it is able to identify. Otherwise, small loci identification is a bit tricky with this approach. Then comes another approach, which is genetic mapping by CNV analysis. CNV stands for copy number variations, and it is applied to the monogenic or monolocous diseases where either single gene is involved or a single locus is there that is causing the disease. So the advantage is that it is the high resolution CNV screening method and no familial cases required, and it can potentially identify the small loci. So in all the previous methods, we have seen the disadvantage that they were not able to identify the smaller loci and they were able to identify the locus which is large. So this drawback is overcome by the CNV analysis in which it can identify the small loci as well. So the disadvantage with this method is that only it investigates the CNVs or the copy number variations. So it cannot detect the balanced events and no base pair resolution. Mutation detection again requires the second step. So in this also it can only target the CNV, it can only investigate the copy number variations, and if there is some balanced event then it won't be able to detect that. So that was the disadvantage with this method. Then came another method, that is whole exome sequencing. So as the name suggests, exome. So exome is a word which is made up of exon plus ome. Ome means when you study something as a whole, and exon means when you study the entire exonic content present at a particular point of time in the human being. So that is known as exome. So in this, you sequence the whole exons. And in exonic sequencing, here they are talking about the exons that are coding for the protein. So it can be applied to any disease, and the advantage is that it provides you base pair resolution exome wide. It detects most types of genetic variations and it can directly identify the causative variant or mutation. So in earlier approaches, you have to perform the second step for the mutation detection. But in whole exome sequencing, you don't have to go for the second step. It itself identifies the causative mutation. It can be applied at the level of exons. It can look for the base pairs changes that happens during a disease. So in that way, this approach is forward than the previous approaches. But the disadvantage with this approach is that it is unable to detect the known coding variants. So I have told you that it is a whole exome sequencing where it is focusing on only those exons that are coding for the protein. So in that way, it is not able to detect the known coding variants, and its limited resolution for the CNVs and other structural variations, and coverage variability due to enrichment process and relatively expensive. So when we move to sequencing level, then the cost that is associated with the approach rises because sequencing is an expensive task. So that's why it is bit expensive and it has limited resolution for the copy number variations or other structural variations. So then came the ultimate method, that is whole genome sequencing. So earlier we were focusing on only the exonic content because what we believe is that a particular disease is only observed in the terms of its phenotype. So we believe that the part of the genome that contributes to that phenotype is the part that is mostly affected. So that's why we were focusing only on the exons. But what we have gathered till now, the information that we have gathered till now, what we have observed that it is not the coding part that is performing the major function, but the non-coding part has also some kind of functionality in the regulation of genes. So if the mutation occurs in the coding part, then it can affect the protein functions. But even if it occurs at the non-coding part, then also it can affect that regulation of the gene or its expression. So in that way, we cannot ignore the mutations that are occurring at the non-coding regions. So in that way, we moved on to the whole genome sequencing level because exome sequencing was not enough for us. So now in the whole genome sequencing, as I've told you, the cost of sequencing is very high. So earlier you were sequencing only the exomes. So they were smaller in size. But when we talk about the genome, then it includes the whole sum of data. So it's very, very expensive. It can be applied to any disease, and the advantage is that base pair resolution genome wide. So it will provide you the base pair information of the entire genome. It detects all type of genetic variations, whether they are occurring at any part, any region, irrespective of coding or non-coding or repeats. It can directly identify the causative variant and mutations. So that is the advantage of this method. This method is the best, but there are certain disadvantages. For example, data analysis complex. So if you are handling such a large amount of data, then its analysis will obviously become very complicated and very complex, and it will be expensive. As I've told you that the sequencing methods are quite expensive as compared to exome sequencing. So these are few of the methods that are generally involved when you go to identify the disease genes. So now let's see two of the main methods. So there are two main methods. One is positional cloning and one is functional cloning. So this is to identify the cause of the disease. For example, a human is suffering from certain disease. Then we can perform these two types of cloning depending on the information that is in our hand. So firstly, we will be collecting the DNA samples from the affected family. Now, if that particular biochemical defect is known for a particular disease, then we'll be going for the functional cloning because we know that what is the biochemical defect. So firstly, let's look into the functional cloning side. So now we have collected the samples and we know the biochemical defect. Now we will look for the protein of known function, that is, so we know the defect, so we will also be having the information of the protein that leads to that performs a particular function which is now missing. So we will be identifying that protein, and now we have the information of the protein. So now we'll move to the gene that is translating through that protein. So as soon as we identify that gene, we will sequence that gene, we will identify the mutation, and these things are done by the comparative analysis. For example, the normal, the organism that is healthy, so we compare his genomic sequence with the gene sequence of the individual who is affected. So in that way, we can identify the mutations. So in this way, whenever the mutation is identified, so we'll be knowing the cause of the disease, that which mutation is leading to that particular disease. So this is very simple and straightforward. That is functional cloning. Now let's see the positional cloning. So as the name suggests, positional cloning, so it can help you to look at the position of that particular disease. So in this, you don't know the defect, that is the biochemical defect that is produced as a result of the disease. So in this, you collect the DNA samples from the affected family, and now you know that you don't have the idea of the biochemical defect. So you check for the chromosomal anomaly in which you will be comparing the chromosomes. You can perform different methods. You can either perform karyotyping. So to identify the chromosomal anomaly, you will be able to see the deletion, that whether there is some kind of deletion of the particular region in the chromosome, or is there some translocation between the chromosomes. So you have also observed the Philadelphia case. So in that there is a translocation. Then you can also check for the duplications. So the copy number variations can also be checked by looking at the chromosomes, by comparing the chromosomes. Then comes the genome-wide linkage analysis methods. So what happened here is you identify the linked locus, you identify the gene at that locus, and whenever the gene is identified at that locus, you will be sequencing that gene, you will be identifying the mutation, and then cause of the disease will be revealed. So in this way, both these methods will be helping us to identify the cause of disease. So here the cause of the disease is identified at the level of the sequence. So you will be identifying the mutations that ultimately cause that disease. So in that way, this will not only yield us the information of the gene that is causing the disease, but it will also tell us about the mutations that are leading to that particular disease, or that are affecting the functionality of the gene in the case of disease. The linkage analysis, as we have seen earlier, that we perform the linkage analysis. So what we mean by linkage analysis, it is, so linkage analysis is a powerful tool to detect the chromosomal location of a disease genes. So we perform it when we want to know about the location of the chromosomes. So means the location of the disease genes in the chromosomes. So it is based on the observation that genes that reside physically close on the chromosome remain linked during the meiosis. So in this, firstly, gene mapping is performed. So for that, we perform pedigree analysis. So families linkage analysis is done. So when this pattern is observed, then we move to the level of chromosomes. And in the level of chromosomes, we try to look for the location of that particular gene, that in which position it is residing. We perform the cloning of particular regions. So after the cloning, genes are identified, And ultimately, after identifying the gene, we move to the level of mutation identification. So this is the method how we perform it. We perform family-based whole genome sequencing. Then if two or more individuals are affected, or affected and unaffected individuals are there, then we exclude those variants that are not shared by the affected individuals and that are present in unaffected individuals. Because here we are trying to compare the variations between the affected and unaffected individuals. So we will be excluding all those variants which are not shared by both affected and unaffected individuals. And then if we have one affected individual, then we will exclude the known coding variants and coding variants that are found in the databases, 1000 Genome Project or XAC, or that are not rare. That means greater than 0.5% frequency. So we will be excluding those known coding variants and the coding variants which are not rare and which are found in these databases. Then we test for the segregation of identified genetic variants with the disease phenotype and ultimately the sequence variants in ethnically matched controls. So in this way, we'll be getting the information of the mutation that has happened and that has finally lead to the disease. Now we know about, as we have talked about linkage analysis. So there are two types of mapping that are performed. One is genomic mapping and one is physical mapping. So as the name suggests, genomic and the physical. So in genomic mapping, we look for the genetic markers and we identify the location of the genes depending on the recombination frequency, that whether two genes will be close to one another, because in this we perform the linkage analysis only to create the genetic map. So we in the linkage analysis we say that two genes will reside to means will be inherited together if they reside close to one another and there are limited chances of their recombinations. So in this way, in genetic mapping, what we does, we identify the location of the genes based on their means how closer or farther they are present. Whereas when we talk about the physical mapping, then in this we map the genes on the basis of their physical locations, in which we use different methods to perform the physical mapping. But in this we get to know about the base pair distance between the genes. So we know exact position or exact location of the genes. We don't have the idea, but we have the exact location as well. So physical mapping is the refined version of genetic mapping. In genetic mapping, you don't have the idea of the exact distance between the genes. You can only say that whether these are closer or not. So now let's look into the differences between the genetic mapping and the physical mapping. So when we say genetic mapping, so it is the process of determining the order of and relative distance between the genetic markers on a chromosome based on their pattern of inheritance. So by performing genetic mapping, we can identify the order and the relative distance between two genes, but we do not know the exact distance between the two genes or the exact position in the chromosomes. And in this, we focus on the genetic markers that are present on the chromosome and we study the pattern of inheritance. Whereas in physical mapping, it is the technique that is used to find the order and the physical distance between the DNA base pairs by DNA markers. So in this way, by using the DNA markers, we look for the physical distance in the between the two genes in the DNA at the level of base pairs. So you can say that two genes are farther from one another by this base pair distance. Then comes the markers. So in genetic mapping, you require the markers such as genes that are genetic markers, and in the physical mapping, the markers are, it could be recognition site of the restriction enzymes. So that is the DNA marker. Then in genetic mapping, these are the maps that depend on the genetic linkage, because as I've told you earlier, that two genes that reside close to one another are meant to be linked and they are mostly inherited together. So in this way, to construct a genetic map, it is depending on the genetic linkage information, whereas in physical maps, you use the visual markers that are short DNA sequences. So genetic maps rely on the recombination and crossing over, because I've told you that we look for the recombination frequency, and means more the genes are more closer to one another, they will be linked and their recombination frequency will also be low. So in that way, it relies on both recombination and crossing over. So if the recombination frequency is low, then two genes will be means they will be drawn closer to one another. Whereas in physical maps, they rely on the DNA sequence of the genome. In genetic maps, they are based on the recombination frequencies, whereas the physical maps are based on restriction digestions. Genetic maps determine the probability of the recombination event between the 2 points. So means if they will be closer, then the probability of recombination event will be less. So in physical maps determine the number of bases between the 2 points. Genetic markers and the size of mapping population are the two important factors. So in this, it involves the fragmentation of the genome either through restriction digestion or physical shattering of the genome. So in this, it is based on the fragmentation by the restriction digestion because it is trying to tell you the distance between the two genes at the level of base pairs. So that's why it has to be fragmented, and then this restriction digestion is performed as it is based on the DNA markers. So the genetic mapping is less accurate, whereas physical mapping is more accurate because it is giving you the relative idea between the two genes, relative distance idea, but it is giving you the exact distance between the two genes, so it is more accurate. Genetic maps provide firm evidence on genetic disorders associated with one gene, for example, cystic fibrosis and muscular dystrophy, or two genes in the diabetes, cancer, and asthma. So in this way, it can provide you the evidences because it is targeting the linked genes or the genes that are far from one another. So in that way, it can provide you the evidence. But physical maps, they are important to identify the origin of the diseases, whether they are inherited or arise due to a random mutation. So in that way, physical maps can provide you this information and they are very important. So ultimately, what is the take home message from this slide is that genetic mapping provides you the relative information about the distance between two genes, whereas physical map provides you the exact distance between the two genes in the terms of base pairs. So in that way, with the genetic mapping, although you can know the disorders that involve one gene or two genes, and if they are linked, then you can, you get the idea, you get the evidence that that particular genetic disorder is associated with those genes as they are linked. So both are affecting a individual for that particular disease. Whereas in physical mapping, you are very confident and you can accurately predict the genes that are involved, as well as you can also accurately predict the mutation, a single mutation, and you can also tell whether that mutation is random or it is not random. So here I have given you the idea about the physical and genetic map. So in this, if you see, this is the distribution of genes based on the sequence data. It is a physical map. Whereas in genetic map, it gives you a rough idea. So you can see that in physical and genetic map, the order in which the genes are placed is exactly the same, but the distance varies. So in genetic map, the distribution of the genes, they are based on the recombination frequency. So higher the recombination frequency, higher will be the distance between the genes. That means the genes are most likely to get recombined. So that's why they are farther from one another. Whereas if genes appear closer together, so it happens when there is low recombination frequency between the genes. And they appear farther apart when there is high recombination frequency between the genes. So there are three kinds of map which I've shown here. One is physical, one is cytological, and one is genetic map. So as we discussed that in the physical map, you know the exact location of the genes. So now I have introduced a new term. Before moving to that term, we look at the genetic map. So it will provide you the exact order of the genes, but the distance that it provides will be relative. In genetic distance, the units which you will use to represent the distance between the two genes is the centimorgan. It is one map unit, it corresponding to the recombination frequency of 1%. Whereas in physical distance, it determined by the DNA sequencing or restriction fragment sizing. So one centimorgan corresponds to 1 megabase per DNA for the human genome. So in that way, these are the terms which are generally, or these are the units which are generally used to represent the distance between the genes in case of physical map and the genetic map. Now, if you see then the cytological map, if you see it appears very thick, it appears as if it is a chromosome. So yeah, physical and genetic map, they are constructed in the single line, but if you talk about cytological map, so it is a visual appearance of the chromosome when it is stained and examined under a microscope. So particularly important are visually distinct regions called light and dark bands, which gives each of the chromosome a unique appearance. So if you will stain a chromosome and you observe inside a microscope, observe under a microscope, then you will see that there will be certain bands appearing. One will be light, some will be dark. If you see here also, the bands are distributed across the chromosome, light and dark bands. So this feature will allow a person's chromosomes to be studied in a clinical test known as a karyotype, which allows scientists to look for the chromosomal alterations. So when we talk about the karyotype, so what map we create is the cytological map with which we track the differences between the two chromosomes. So in this way, these are the different types of maps that are developed. So here I've given you the example of the genetic mapping. For example, if you want to calculate the recombination frequency, then it is calculated by the formula. The formula is the recombinants divided by the total offsprings multiply the 100%. So you will be given the numericals in which you will be provided the number of recombinants and the total offsprings that are present, and then you will have to find the recombination frequencies. So for example, in this case, the recombination frequencies, so there are three genes that are present, A, B and C. So recombination frequency between A, B, BC and AC are given. So in this you can see the largest recombination frequency is 18.5 between A and C, which means higher the recombination frequency, higher will be the distance, because now it can easily recombine. So that means they should lie farther from one another. So in this we can see clearly that A and C will lie farther from one another. Now if we look at AB and BC, we can see that A and B are farther, its recombination frequency is high, whereas BC has comparatively lower recombination frequency. So B should lie closer to C and farther from A. So in this way, here we can draw B. So in this way, it is a genetic map that we can create based on the recombination frequencies. So I hope it is clear. So now let's move to the another topic, that is physical mapping. So how do we construct a physical map? So this is one of the technique which is based on the restriction digestion. So what we does here is we take the cells that are required to collect the genomic DNA. So firstly the cells are lysed, they are broken to retrieve the genomic DNA. Then single genomic DNA molecules are placed into the microfluidic device, and in this device we provide the restriction enzymes so that they can cut the DNA molecule at specific recognition sequences, and each DNA molecule is now stained with the fluorescent dye. An optical map of the single molecules are derived by measuring the fluorescent intensity. So in this way, now the fluorescence intensity will be measured for different fragments as they have labeled individually. Each fragment is labeled individually. So now in this way, we can construct a consensus genomic optical map. So the overlapping of the multiple single molecule map gives us the consensus genomic optical map. So at last, we will be getting the entire genomic map by performing this restriction fragment digestion. Then there is another method, that is FISH, that is fluorescent in situ hybridization. So in this you have the information of the gene, you cut it with the DNAs and you perform the nick translation, and where the nicks are, so nicks are nothing but nick is known as the, you can see here the cut that is present. So that cut, when you create some when you remove some nucleotides, and the cut that is produced is known as nick. So nick translation means when you translate, then when you fill that nick, so generally it is filled with the um, by means labeled um, CTP, ATP nucleotides, triphosphates. So in this way it is constructed. Now there is a fixed cell on the slide which contains some DNA. It is denatured. Now we perform the hybridization so that this DNA, or means the sequence that is specific to this gene, could bind this particular DNA that is obtained from the gene. So wherever it will find its complementary sequence, it will bind specifically to that region, as we have labeled it with specific probes. So now we will add some complementary molecules such as antibodies which could bind these biotin DUTPs, and we will also add anti-dig, that is avidin linked with the fluorophore. So now this fluorophore will be visible when we observe it under the epifluorescent microscope. So in this way it can tell you that whether that gene is present in that given DNA sample or not. So it is a genomic sequence that has obtained from a particular cell, and this is the gene that you are Means you know that this you want to identify this particular gene within that. So in this way you can specifically label that region in the genome, and you can identify that whether that gene is present. If the fluorescence is observed, that means the gene is present, as this complementary sequences bind properly, and this antibodies and fluorophore has also attached. So these are the two main methods which we use for the physical mapping. So now let's look at the complete difference between the genetic and physical mapping. So we have already discussed, but we'll just summarize this. So in genetic mapping, it depicts the relative position of the loci depending on the degree of recombination. Therefore, it studies the inheritance or assortment of traits by the genetic analysis. So hence, the types of markers used in genetic mapping is the genes. Now, in contrast to genetic mapping, if we look at the physical mapping, then it is another technique that determines the actual distance between the loci using the number of nucleotides. For that, it uses the technique of molecular biology, such as restriction digestion and DNA sequencing. Also, the restriction recognition sites serve as the DNA markers for physical mapping. So, on that account, the main difference between the genetic and physical mapping is the type of markers and the techniques that are used in mapping. Genetic maps provide firm evidence on genetic disorders associated with one gene or the two genes. On the other hand, physical maps are important to identify the origin of the disease, whether they are inherited or arise due to random mutation. So, this is all about the genetic and physical mapping, and I hope now it's clear that what is the difference between these mappings and how these mappings are performed and what advantage they offer to us and when to use which kind of mapping. So now let's move further. So now we were discussing about these methods where we are going to identify the disease genes, where we were targeting the sequence and we were identifying the mutation. So now what happens if a particular disease is not caused by just one gene or two genes, but multiple genes? And there could be multiple sites that are causing that disease. So such diseases are the complex diseases. So they are defined as the disorder that results from the contributions of multiple genomic variants and genes in conjunction with the significant influences of the physical and social environment. So these are the disease which arises due to the contribution of multiple genomic variants. So here you can see there is a single gene disease that is arising due to the defect in the single gene, or there is single variation that causes that disease. But in the left you can see the complex disease. These are the multiple variations that are at the same time causing that disease. So many genomic variants are the polygenic interacting with the environmental factors. So when we look at the monogenic disorders and polygenic, so in polygenic we say multiple genes are involved. So this is the way in which multiple gene variants are causing a disease. And other than this, environmental factors also play a vital role in the disease. So here they have given the example of coronary artery disease. So here you can see in all the chromosomes, so 23 pair of chromosomes, in every chromosome there is some variation lying. So these are the complex diseases. Whereas in the sickle cell anemia, you know that there is a single variation and that is present in chromosome 11. So these are very easy to detect, but the complex diseases are complicated. So in Mendelian disorders, they are generally tested for the single gene disorders. Standard genotyping is performed. We confirm or rule out the disease based on the extent of testing, and we treat and manage the disorder using the population statistical evidence. Whereas in complex disorders, we test for the common risk variants. For example, we check for the SNP arrays or NGS, which is next generation sequencing. We classify the complex disorders using the disease models, genetic and environmental risk factors, and disease biomarkers. We determine the mechanistic defects, disease state, and predict the clinical course. So these are determined and these are predicted. So manage the disorder using the patient-specific targeted therapies aimed at preventing the progression and adverse outcomes. So in this way, the complex disorders are dealt, and these are far complicated than the Mendelian disorders. So the approaches which we have discussed earlier, they were targeting the Mendelian disorders and they were based for Mendelian disorders. But now that as the disease has complicated, they have become complex. So now we should target different variations. So then comes the new concept. So now let's see that how these strategies have evolved. So linkage strategies, if we look at the traditional strategies which have arrived at 1980s or earlier, so the linkage analysis on pedigrees, association studies such as candidate gene approach was used, allele sharing methods, affected siblings were studied, then animal models were used to identify the candidate genes and cell hybrids were created. But then from the 1990s, newer approaches arrived which focused on the special populations, then haplotype sharing, because in the complex disease, what happens is multiple variations are causing a particular disease. So we must look for the haplotypes or the variants. So haplotype is nothing but it is the grouping of all the SNPs together. We will look into its detail in the further slides, but as of now, you take this idea that in this we check the variants and many variants, not just a single variant, but we are targeting multiple variants. And the techniques that are on the horizon are single nucleotide polymorphism and functional analysis, where we find the candidate genes. Now, if we look for the future, then there are certain techniques which are much needed. For example, new multi-locus analysis techniques, because all the approaches or techniques which we have discussed, they were targeting a single locus. So we require the techniques that can target multiple locus and ways of dealing with the larger pedigrees, so that if a larger pedigree is given, so we require certain approaches that could deal with the larger pedigrees and better phenotypes, one closer to the gene products, and then we require the larger collaborations. So these are needed and the work is still going on these approaches. So with each time the approaches are being updated. So now in few time we can see new approaches coming up, but now let's discuss about the variations, because we have studied the complex diseases. So now let's see what are the variations. So one term is that you have heard just now is the SNP, that is single nucleotide polymorphism. So what does SNP means? So it is a genomic variant at a single base position in the DNA. Scientists study if and how SNPs in a genome influence the health, disease, and drug response and other traits. So as its name suggests, single nucleotide, so it means the polymorphism that arises due to the change or the variation of the single nucleotide. So here you can see these are the four individuals, and this particular region is the SNP, that is single nucleotide polymorphism, which varies among these four individuals. So in maternal, you can see here is T, whereas in paternal at the same site C is present. So there is a variation that is present, which will lead to a polymorphism. So in all the individuals, this site shows some variation. For example, here maternal and paternal have the same, but if we compare individual 3 with individual 2, then it is entirely different. This is C present here. So in this way, when we say SNP, the polymorphism, we talk about multiple individuals, and different individuals, different nucleotides are present, different sequence patterns are present. So that is known as the polymorphism arising due to the single nucleotide. So that is regarded as SNP. Now we were saying haplotype. So what does haplotype means? So haplotype is a physical grouping of the genomic variants or polymorphisms that tend to be inherited together. So in complex disease, we say that multiple variations are there. So when we talk about SNP, then we talk about the single nucleotide polymorphism, a single site. There could be multiple SNPs in the genome, but when we say SNP, we only talk about the single position. So when we group all those SNPs together and we construct a sequence that involves all the variations or the unique combination of the variants that are present inside the chromosome, then that is referred to as haplotypes. So here you can see these are the three individuals and these are their genomic sequences, whereas these are their SNPs. So in the haplotype, we have considered the information of all the SNPs and not the other regions. So one of the reasons that why we are uh, doing this, why we are refining our data set, why we are means from genome we are moving to the haplotype, we are targeting SNPs. So this is done because the cost of sequencing is very high and analyzing the larger sequential data is also very complex. So we try to reduce the data set by removing all those regions that might not be important. So in this case, if we want to compare the two individuals, then the regions that are exactly same between both the individuals are none of our interest, and we only want to see that where are the variations occurring and how those variations are causing the disease. So in the haplotype, you are only focused on the variations, variable regions. So in that way, you don't have to deal with the entire genomic sequences. You only have to deal with the variable sequences, and in this way, the analysis will become quite easy as compared to the previous methods where the entire genome sequence was targeted. Now we say haplotype and genotype. These are two terms. So why we are looking for the haplotypes and not the genotypes? So in haplotypes, what haplotype means? It is a grouping of the SNPs together. So in haplotype, haploid, the name also suggests haploid. So it is a single set, single sequence from the single individual. For example, here I have shown you these are the haplotypes. So in this, if we talk about individual one, so maternal and paternal sets or the sequences were given. So if we have to look for the haplotype, then it will only be coming from the one of the parent or the single copy. Whereas if we talk about the genotype, then it involves both the copies coming from both the maternal and paternal chromosomes. So in this way, genotype will be the in terms of pair, whereas haplotype will be the single copy sequence. So these are the haplotypes from different individuals. So whenever we want to draw the relationship between the phenotypes and the variations that are leading to that phenotype, then there are different criteria which must be satisfied. For example, in the phenotype here they have given the lung cancer and no lung cancer. For example, you have given these different genotypes of the individuals and you have to say that what variation is causing this disease. So in that you have to compare their genotypes and you have to identify the variation that has arrived, that has caused that disease. So if we look at the haplotypes and the genotypes, if we compare them, then we will see that for a particular haplotype or a particular sequence to be involved in a particular disease phenotype, then it should be shared by all the affected individuals. So here you can see this red boxed sequence is shared by all the three individuals, 4, 5, and 6, whereas it is absent in the individuals that are healthy, which are not showing any lung cancer. So first of all, for any sequence to fall in the disease category, it should only be present in the individuals who are diseased and it should be completely absent from the healthy individuals. So first criteria is that, which is satisfied here. But whenever you look for the genotypes, then you will see that the sequence of this individual, of individual 6 and individual 3, is exactly the same. So now how you will predict that what is the variation? And the another criteria which must be satisfied is that all the individuals that are affected should share the similar sequence which is causing the disease in one. So it should cause the disease in another one also. So first of all, a sequence should only be present in the individuals that are affected and it should not be present in the unaffected. And another thing is it should be exactly the same in all other affected ones. Whereas if you see here, then all these three, their sequence appears different, but their sequence should share some kind of similarity because they all are affected. But when you go at the level of genotypes, then the identification becomes complicated because it is looking at the pairs. So it's a bit tricky to identify the variation that is involved. Whereas if we look at the haplotypes, it is very easy to identify the region or the sequence that is involved in that disease. And in that way, ultimately, you can look at the variation which are involved with the particular phenotype. So now we can say that the haplotype analysis is better as compared to the genotype analysis if you want to specifically know about the variations related to a particular disease. So now human genome project. So because to check for the variations, we require the information of the sequence of the genome. So for that, human genome sequence was proposed. So these shows all the landmark events that happened in the genetics and genomics and how we further moved to the level of human genome and how we sequenced. So this begins from the discovery of DNA, its structure, then with the Mendelian inheritance, then further with the different recombinant DNA methods, the sequencing methods, the electrophoresis methods, they all came up. So then came Human Genome Project in 1990s. It continued till 2003. So these are the major events that happened during the Human Genome Project. So as the name suggests, Human Genome Project, so don't get confused. It was not only means designed for sequencing the human genome, but it was trying to sequence the other organisms as well. But ultimate aim was to sequence the entire genome of the human. So in the midway, you can see that various other genomes were sequenced of different other individuals which are simpler than humans, such as mouse, C. elegans, E. coli, Drosophila, housefly, mice. So in this way, ultimately at 2003, human genome sequence was completed. And now you got the human genome sequence, so now you can analyze it, you can compare it. And human genome sequencing was very much expensive and complicated. That's why it took so much time. So now, as the genome is sequenced, we know about the number of genes that are present. So then came the another project, that was International HapMap project. HapMap means haplotype map. It is a short form. So HapMap project came because to deal with the entire genome sequence is very complex. If, as I've discussed during the haplotype introduction, so we said that why we move to the haplotype level, because if you deal with the genomic DNA and then we individually compare the SNPs, then it's very difficult and it's very complex because you have to deal with the such a huge sequence. So that's why haplotypes were constructed. So similarly, after the Human Genome Project, as human genome was sequenced, so our next aim was to look for the variations that are present in the genome. So that's why this International HapMap project came up. And this project will also simplify the analysis because now we don't have to deal with the entire DNA sequence. We will simply deal with the smaller chunks or the smaller variations. So this will provide us better results. So in this, they say that the elucidation of the human genome has made it successful or made possible our current efforts to develop a haplotype map of the human genome. And this haplotype map is referred to as HapMap, and it allows the researchers to find the genes and genetic variations that affect the health and diseases. So the DNA sequence of any two people is 99.5% identical. So as I've told you earlier that if you want to compare the genes that are causing the disease, so you will be comparing only those regions that are variable, because the rest is identical. So identical regions will perform same functions. So no need to look at those regions. Similarly, they are saying that if you are comparing two people and if 99% of their DNA is identical, then what is the means motive? Why to look at that if you know that it is identical? So in that way, this HapMap project was developed, keeping that thing in mind. So the variations may greatly affect an individual's disease risk. So sites in the DNA sequence where the individuals differ at the single DNA base are called the single nucleotide polymorphisms, which we previously discussed. And when we create a set of these nearby SNPs on the same chromosomes, then that is known as haplotype, and it is generally inherited in the blocks. So means if two SNPs are lying together or lying closer to one another, then they will be inherited together. Now they say that this pattern of SNPs on the block is known as haplotype, and blocks may contain large number of SNPs, but few SNPs are enough to uniquely identify the haplotypes. So now this HapMap is also constructing the haplotype map, all this NP information. So now they have said that you don't require all this NPs, even if you have the information of the means um, few SNPs, then also it is enough. So in that way, HapMap is the map of these haplotype blocks, and the specific SNPs that identify the haplotype are called tag SNPs. So now they have further refined it. Now they are not dealing with all the SNPs, but they are dealing with the few SNPs that are sufficient to represent the haplotype, and they have called those SNPs are tag SNPs. So in this, this HapMap will enable the geneticists to take the advantage of SNPs and how the genetic variants are organized on chromosomes. Genetic variants that are near to one another are tend to inherited together. So in that way, these are the different ways, these are the different information that why HapMap project was developed, what is haploid map, and it contains all the SNPs, and how you can see how we are moving further, how we are concentrating our methods, means with the further, in starting with the huge data set, we are refining it further and further so that we could analyze it properly. So in this also, we move to the level of tagged SNPs, and we said that SNPs which are residing closer to one another tends to get inherited together. So this SNP, this linked variants, this phenomena is influenced by the recombination and linkage disequilibrium. So recombination can break that. Means if two SNPs, either they are located too much closer to one another, but if recombination happens, then they won't be inherited together, they get recombined. So in this way, the two processes that can affect this linked variants are the recombination and the linkage disequilibrium. So here I've given you the example that the HapMap project, how it has enabled the means geneticists to take the advantage and check that how the SNPs and other variants are organized on the chromosome. So this thing we have already discussed, that it could be influenced by the recombination linkage disequilibrium. So in this they have given you the example where they have given you the 4 chromosomes, and these are the SNPs that are present in these chromosomes, and in the haplotype all the SNP information is collected and it is kept. Now from these SNPs, only the 3 SNPs have selected which are sufficient to represent a haplotype. So that is known as a tag SNP. And here I've provided you the definition. So a tag SNP is a representative single nucleotide polymorphism in a region of the genome with the high linkage disequilibrium that represents a group of SNPs called a haplotype. So in that way, tag SNP is nothing but it is a representative in the region of genome, and it has higher linkage disequilibrium, and it represents the group of SNPs. So means if two SNPs are have high linkage disequilibrium, so linkage disequilibrium is nothing but it is also another measure which tells you that what is the probability if one gene is present, and what is the probability then that another allele will also be inherited. So it means if the means segregation of the two alleles is dependent on one another, or it can, the probability of assortment of the two alleles are means very dependent on one another, then that is linkage disequilibrium. Because we have also discussed it previously and we will also discuss it later, that what does it mean and how it is different from linkage equilibrium. So don't get confused. As of now, you should only remember that linkage disequilibrium is a thing where the two alleles are linked to one another and they always lie closer to one another, they are linked and they get inherited together, and their frequency of recombination is very low, or you can say it's zero. So that is linkage disequilibrium, that their inheritance is dependent on one another. So now let's practice few MCQs as we have discussed a lot about the content. So now we will talk about MCQs. So first MCQ is the ability of two mutants in combination to restore a normal phenotype is known as. So now you have to tell that what is that ability known as, where the two mutants in the combination they restore a normal phenotype. So option A says hybrid vigor, option B says complementation, option C says backcross, and option D says anticipation. So you can think and then you can answer. What do you think? You can comment or you can chat. You can type your replies in the chat or you can unmute and speak up. Okay, so few people are saying it's option 2. What about the rest? What do you think? Okay, so let's see. So its correct answer is complementation. So we have previously discussed about this, that means how the two mutants when they combine then they restore a normal phenotype. So this is known as complementation. Whereas if we talk about anticipation, so in that also we have discussed that when the repeats exceed beyond the normal range, then it becomes disease, the organism become diseased, risk factor increases, so that was anticipation. whereas backcross was performed when you want to, uh, you know, you perform with the recessive parent. So that is just to purify the progeny. That was the backcross, and then hybrid vigor. So means hybrid vigor is nothing but it is the means it is also known as heterosis. It means that if there is increase in the biomass, fertility, in the stature, when there is some better version is produced, so that characterizes the progeny of crosses between the diverse parents, such that F1 is superior to the better of the two parents. So whenever some better offspring is produced than the parents, then that was hybrid vigor. So in this way, the term which represents the ability of two mutants to restore a particular phenotype, so that is known as complementation. So the next question is the genetic disorders with anticipation. So in the previous also I've told you about anticipation. There was involvement of repeat sequences. So the option says involves repeat expansion, affect both sexes, show severity of the disease inversely correlated with age at the onset in successive generation, or all of these. So what do you think when we talk about genetic disorders with anticipation? So what does it involve? Because I'll give you the example of Huntington's disease, which we have previously discussed. So in that also we have seen that how a particular region, how a particular trinucleotide repeats beyond a particular range or beyond a particular number. If that repeats, expands, then it causes the disease. So you can answer in the chat. Few of the students have answered option 4. What about rest? This was in your assignment 4th, I guess. These results are already out. Okay. So maximum have answered option 4 as the correct option, and it's true. So genetic disorders with anticipation, they all involve repeat expansions. They can affect both the sexes, and it shows severity of the disease inversely correlated with age at onset in successive generations. So in the previous session, we have discussed that how the mechanism, what was the mechanism that how these repeat get expanded. If there is some slippage during the replication, then what happens is these repeats get expanded, and with each further replication round, this number will keep on expanding. So in that way, in the successive generations, now this disease onset will be quick. It will begin at lesser age and it will be more severe. So in this way, this line says that it is inversely correlated with age, the severity. So that means if the age will be less, then severity will be high, and that will be in the successive generations. So this is all about the genetic disorders with anticipation. Now let's move to the next question. So the next question says, in polygenic disorders, the term genetic risk factor refers to. So we have discussed about polygenic disorders and the complex diseases. So in this, what happens is multiple genes are involved. So they are saying that when we say genetic risk factor, so what does that mean? Option A says a gene mutation that confers a 100% risk of developing a disorder. Option 2 says the XY or XX genetic constitution influencing the gender-specific disease onset. Option 3 says a gene variant that confers a small yet significant risk of developing the disorder. And option 4 says a known genetic factor that confers a small yet significant risk of developing the disorder. So what do you think? What does genetic risk factor means? Okay. So many students have replied in the chat. Okay, so let's see. So many have answered option C. So yeah, it's right. When we say genetic risk factor, so it means a gene variant that confers a small yet significant risk of developing the disorder. So some were confused with option 1. So in genetic risk factor, it is not always that it will always give you 100% risk. It could it confers a small but significant risk of developing the disorder. So in that way, option 3 is right. Now let's move to the next question. So finding the defective gene for an autosomal recessive disorder using a conventional approach is a sequential process. Identify the option that correctly orders various steps in this process. So now you have to tell the correct order. So we have already discussed the how we will identify the disease genes. So I think it might help you with this question. So they are giving you the order that how we perform the analysis for the defective gene. So first option says firstly gene candidate, mutation analysis, physical mapping and segregation analysis. Option B says positional gene candidate, mutation detection, physical mapping and linkage mapping. Option C says linkage analysis, physical mapping, homozygosity mapping, and positional gene candidate and mutation detection. And option 4 says shotgun sequencing, pedigree analysis, mutation detection, and positional gene candidate. So now what do you think? What will be the approach? What will be the order? From which order we have to move further? Okay. Few students have answered option 3. Okay, let's see what is the correct answer. So yeah, you were right. Option 3 is the correct answer. So firstly, we'll begin with the linkage analysis. Then we will perform physical mapping, where we will look at the distance in the terms of base pairs. We will check the homozygosity mapping. And then positional gene candidate, we will know what is the position of that gene. And after getting the positional information, we will be detecting the mutations. So in this way, we are performing this homozygosity mapping here because it is a autosomal recessive disorder. So in this way, by performing all these steps, ultimately we will be getting the information of the defective gene that is involved at the level of sequence, that what kind of mutation is involved. So let's move to the next question. So this question says, a newly married, phenotypically normal female individual was found to have a balanced translocation in her karyotype. Which one of the following option is incorrect regarding this condition? So now they are saying that female is normal and there is balanced translocation. So in the previous session also many students have asked me that ma'am, if there is balanced translocation, then will it cause any disease or will it be safe always? Or if there is imbalanced translocation, then it is always harmful, or sometimes it could be silent, it could not affect an individual. So with this question you'll get a bit clarity about this. So let's look at its options. So it says option A that higher chances of recurrent abortions. Option B says she carries a fusion gene resulting from the translocation. Option 3 says the balanced translocation will not have an adverse effect on her survival. And option 4 says the translocation did not affect the function of any of these genes. So if you look carefully, you have to select the option which is incorrect in regard of this condition. So what do you think? Okay, few students have mentioned option 1 in their answer. Okay, few students have mentioned option 2 as well. Okay. So what about the rest? What do you think? You can guess. Okay, then let's see. So the correct answer is option 2. So what happens is that there is balanced translocation. First of all, we'll discuss with this option. So in this, they are saying that she carries a fusion gene resulting from the translocation. So first of all, it is a balanced translocation. Second, it is occurring between the homologous chromosomes. So by it is not possible that there will be the generation of fusion gene from translocation. So this is completely incorrect. Now let's look at the another options. But first of all, let's look at the reasoning. So what it says that a balanced or chromosomal translocation is a condition in which part of the chromosome has broken off and reattached in another location without affecting the function of genes that are located close to breakpoints. However, such individuals are at higher risk for miscarriage. So here I think you have got the clarity that there will be no fusion and no gene function will be affected, but it could affect the individual or the offsprings. So it will enhances or it will increase the risk of miscarriage. So in that way, higher chances of recurrent abortions is right regarding this balanced translocation. Then the balanced translocation will not have an adverse effect on her survival. So it's true because means mother will not be affected in which the balanced translocation has occurred. So normal female is normal and balanced translocation will not affect her survival. And the translocation did not affect the function of any of these genes. So this is also clear that wherever the breakpoints are there, no gene function will be affected. Genes will not be broken off. So in this way, all these three options were, true regarding this condition, but this one is the incorrect one. Okay, let's move to next question. So this question says you have been given purified biomolecules from a rare species of wild bat and you are asked to identify the number of exons in one of the genes OZ involved in the olfaction, assuming that OZ gene does not have orthologs in other species and that it is a novel gene and that you have the ability to sequence the biomolecules. Which one of the options availability of biomolecules is likely to help you define the exons for the OZ gene? So in this, they have given you the option that which two biomolecules you can use and you can identify the number of exons. And this is the gene. And then they have given you a few hints that this gene is not present in any orthologs. It is novel gene. So what do you think? How you can check the number of exons? So first option says mRNA and protein. Second option says genomic DNA and protein. Option C says genomic DNA and mRNA. And option 4 says any of these. So what do you think? If you want to get the information of exons and you also want to tell the number of exons as well, you can think and you can answer. Okay, few students have answered option 3 and some are confused with option 1 as well. Okay, so now let's see what is the correct answer. So the correct answer is option C, which is genomic DNA and mRNA. So now I'll explain. So why genomic DNA and mRNA and why not protein? So first of all, you must know that when we talk about protein, then what protein involves? It involves all those exonic sequences which are coding in nature. And when we were discussing our assignment 2, then we saw that there are certain regions of the exons which fall into the UTR regions, which are untranslated. Before the start codon, all the sequence of the exon is not coded into protein. So first of all, you can't use protein because if you use the protein, you will mess up with the information of exons that are lying in the UTR regions. So that's why we cannot use protein here. We use mRNA because it gives you information of all the exons, and here they have said mRNA. So when it is written mRNA, it means it is a mature RNA, mature mRNA, which means in this splicing has already been occurred, introns have been removed. So to calculate the number of exons, you must have idea about the introns because in mRNA you see all the exons are combined together and it's very difficult to check for the boundaries or the borders of the exons. So to get the idea of the boundaries or the border of exons and to check for the number of exons, you have to compare that mRNA with the genomic DNA. So when you will perform the comparisons, because this genomic DNA will have both exons and introns, so if we will compare the exons, means mRNA with the genomic DNA, then we will know that where that exon is lying in the genomic DNA, and then we will be counting the number of exons. So in that way, the option 3 is the right answer. In this, they have given you the explanation that the exonic sequence harboring the untranslated known coding regions can be identified by comparing the sequence of mRNA and genomic DNA. Protein does not provide information on known coding exons. So yeah, there are certain exons that remain as known coding for the protein. So In this way, we are using mRNA and genomic DNA. So I hope it is clear. So certain students have asked me the doubt, and they were confused that what is orthologs, and they were also confused about the novel genes. So let's see. And one more question somebody has asked, that what is the difference between the orthologs and the homologs? So here I have given you the entire information. I'll tell you. We will be, and I'll be explaining this. You will come to know. So when we say homology, so homology is a bigger term. Orthology and paralogy are the subterms which comes under the homology. So homology means when we say that a particular is a descent from a common ancestor. When there are two individuals that have arrived from a common ancestor, or there are two genes that has arrived from the common ancestor, then those genes are referred to as homologs or homologous genes. So the definition says that the homologous gene or the homologue is a gene inherited in two species from a common ancestor. For example, if there is a species of human and monkey, and there is some gene that has come from mouse. So although the sequence can be a bit different, it could be similar. So it is coming from the common ancestor. So mouse is a common ancestor from human and monkey. So if that gene is inherited from the mouse, so that is known as homologous gene. That means it is inherited in two species from the common ancestor. And while homologous genes can be similar in sequence, and similar sequences are not necessarily homologs. So this is a major term. They are saying that for any gene to be homologue, it should come from the common ancestor and it should share the sequence similarity. But if two genes are showing sequence similarity, then it doesn't mean that it is a homologue. So the main condition for homologue is that it should have the common ancestor from which it is coming. And now there are two other terms which you must know, which you should not confuse. One is similar, one is identical. So when we say similar in sequence, then we mean that there is similarity. For example, if we write two protein or two gene sequences, so what happen is if we talk at the level of proteins, then there are certain amino acids which have similar nature. Or if we talk about the nucleotides, then there are certain nucleotides which are almost having the similar nature. For example, the purines and the pyrimidines. So in this perspective, if we talk about the similarity, then we talk about the similarity at the level of protein sequence of the amino acids. So two amino acids, for example, there are certain amino acid categories such as polar, non-polar, charged, so positively charged, negatively charged. So if in the two gene sequences, if there is variation between a sequence in which one amino acid is replaced by the another, but the nature of both the amino acid is same. So at that condition, we will consider both the sequence as similar. But when we say identical, so we means exactly same. For example, if valine is present, then in another also valine should be present. If asparagine is present, then in the another sequence also asparagine should be present. If glutamic acid is present, then in another also glutamic acid should be present. But when we say similar, so it means that even if in a one sequence glutamic acid is present and in the another if aspartic acid is present, then also they will be considered similar. So this is the difference between the similarity and the identity. So in this they are saying that they can be similar in sequence. So similarity means not entire sequence is identical, but there could be certain variation that could lie, but that variation should be like minor variations. The nature of the amino acid should remain the same. And then they should have been coming from the common ancestor. Then only we'll regard it as the homologues. Now homologue can be divided into two terms further. One is orthology and one is paralogy. So in orthology, what we means the descents from the common ancestors by the genomic divisions. So when division has occurred, then this particular gene was transferred to two different species. So this is known as orthology, and these two genes are orthologous, and the organisms that contain these genes are known as orthologous organisms, yeah, orthologs. Whereas the paralogy is the descent from the common ancestor by duplication within a genome. So, for example, there was some gene present in the ancestor, and now the organism that inherited that gene has further undergone the have shown the duplication of that gene, now there will be two genes. And with each round of, means when evolution will happen, then what happen, these two genes will become bit different in their sequence, but still they arise due to the duplication and they also inherited from the ancestor. So that's why it is regarded as paralogy. That means there has occurred some duplication. So we can see the definition. So orthologous means, orthologous are the homologous genes where a gene diverges after a speciation event. So speciation means a new species has arrived, but the gene and its main function are conserved, but the functionality has remained the same. Conserved means even if the speciation has happened, even if the new species has arrived, but the function is the same. So for example, if there is particular gene that was present in mouse and it was performing some function such as kinase, so it was performing the kinase function, so in human also it will perform the similar function, so that is known as orthologs. So if a gene is duplicated in the species and the resulting duplicated genes are paralogs of each other, even though over time they might become different in the sequence composition and function. So here, if we talk about orthology, then we talk about two species, but if we talk about a single species only, we can also see it in the others as well, but what happens here is that in the single species only, that single gene has duplicated. So now it's means if you see that one gene has undergone the difference, but if you will compare these two, then also they'll appear a bit similar. So in that case, these two are not orthologs, but they are paralogs of one another, because that has arisen as a result of duplication of the ortholog gene. So this is the main difference between the orthology, paralogy, and how the homology is the major term or the major group. So I hope now it is clear that what does the orthologous gene meant in the question, in which they said that the uh, this OZ gene does not have orthologs in other species. So it meant that the OZ gene was not inherited from some common ancestor because there are no orthologs present. So this gene is novel. Now let's see what does novel mean. So when we say novel gene, so a gene that is only found inside a single species or a branch but not outside, and it is also regarded as orphan in that specific branch. So novel genes are classified by their age. It means genes that have emerged inside a defined time frame are the novel genes. So, in simple words, you can say the novel genes are the genes that are only present in that particular species. They have not come from any ancestral species. You only have observed that particular genes in that individual which you are studying right now. So, how means you can see that evolution has occurred. So, how does normal genes came? So, they might have come from some variation, because so now these are different factors, or you can say different reasons that how the novel genes arrived. So first is the duplication and divergence. So what has happened that the primitive gene that was already present, so it has duplicated or and then it has diverged, due to which it became novel. So now in the new species it has observed, whereas in the primitive species it was not present. So it arises due to the duplication and then divergence. Then comes gene fusion. For example, in your ancestor, two genes were present, but now in the current organism, those two genes have combined together and now a new gene is produced. So this is also a gene fusion. Now another reason is that if there was single gene in the ancestor and now this gene has divided into two genes. So if it get divided, there is a fission in the gene. If one gene split into two genes, that is also the reason that two novel gene will be observed. Now the another is horizontal gene transfer. So horizontal gene transfer means if it is not transferred vertically, that means if some virus has infected an individual or if some bacteria has infected, so what is happening is that that is externally injected, or that is externally passed on to an individual. So that is horizontal gene transfer. So when we talk about vertical gene transfer, so when we say the parents give rise to the offspring. So in that way, mother and father both transfer their genes to their offspring. So that transfer is known as vertical gene transfer. But the horizontal gene transfer is that it is not from parent to offspring, but it is the transfer that is occurring due to the external agents such as virus infection, or some vectors, or some external agents. that are artificially or that are means passing on that gene. So in bacteria also it was observed. So now comes the another reason is the retrotransposition. So retroposition can also do this. Novel gene can also be formed as a result of retroposition by changing the positions. So okay, now let's move further with another question. So now this question says Angelman syndrome, an imprinting disorder, was diagnosed in a 1-year-old boy Dinesh, and his mother Lata was found to be the carrier for the mutation in the UBE3A gene. Lata's brother Ratnesh had recently married and was expecting a child, and he had also been diagnosed as a carrier. What is the probability that Ratnesh's child will develop Angelman syndrome? So these are the four options that are given. So as you already know that this Angelman syndrome, it occurs as a mutation in this particular gene, and you must remember that Angelman syndrome is the syndrome in which there is maternal imprinting, in which the copy of the mother's gene get imprinted or get silenced. So in this they are saying that this there is particular mutation in this gene. So Lata was the carrier for that mutation, and when she gave rise to her child, then that child is also detected with that particular syndrome. So now Lata's brother Ratnesh, who has also recently married, so now they are saying that he has also been diagnosed as a carrier. So now, what is the probability that his offspring will also be affected? So what do you think? So now this Ratnesh is also carrying that particular mutation, and you know it is a maternal imprinting. Okay, few people have replied in the chat as option A or 1, 0%. What about the rest? Okay, so option 1 is correct, which means 0%. So one thing you must remember that in Angelman syndrome, it manifests only when the disease allele is passed by a female or the mother to the next generation. But one thing you must keep in mind that it doesn't mean that the this Ratnesh will not pass this. Ratnesh can pass this to their child. That child will be a carrier. So when we say manifests means it get affected. It shows phenotypic expression. So in this, they are saying that it only manifests when it is passed by the female. But you must keep in mind that father can also pass it, but in that case, the offsprings will only be the carriers. They will not manifest the disease. They will not show the Angelman syndrome. But if they further means if they further have the child, if for example, if father pass on this to his daughter, and then daughter will be a carrier. But if that daughter further um, give means marry someone and give rise to offspring, then that offspring will be affected. So in that way, this thing you must keep in mind that it manifests only when it is passed by the female, but it doesn't mean that male won't pass it. Okay. So now let's look into the details. So in Angelman syndrome, it is a genetic disorder. It is a problem which occurs as a result of the gene deletion, which is the UBE3A gene, which is ubiquitin protein ligase E3A present on the chromosome 15. When there is some problem in this gene, either this gene is silenced or this region is deleted, then this Angelman syndrome happens. So it is a missing or defective gene that is the factor of this syndrome. So you receive your pair of genes from your both of your parents, one copy from mother, one copy from father. And your cells use information from both the copies, but in small number of genes, only one copy is active. So this maternal copy UBE3A gene is active in the brain. So what happened here is that now this male copy is inactive in the brain, but this female copy is active. So only this gene can express in the brain if it comes from the maternal copy. Means if mother is giving this gene, then only it get expressed in the brain, and the paternal copy is transcriptionally inactive. It won't express. So in most cases, Angelman syndrome occur when the part of maternal copy is missing or damaged. So that's why it is known as maternal imprinting. It is only happening when the maternal copy is silenced. So because maternal copy can only express in the brain and paternal copy can't express. So whenever this syndrome happens, then it is mainly a neurological, it shows neurological effects because now no copy is expressed in the brain. So that's why the syndrome occurs. So in this way, in few cases, Angelman syndrome is caused when the two parental copies of genes are inherited instead of one from each parent. So you can also see that even if it is not a disomy case, even if paternal disomy is not observed, even if you see both maternal and paternal copy, but still if Angelman syndrome is present, it means the particular region, that is UB3A gene region, is silent in the mother's copy. So that can also be possibility. It is not that maternal copy will never get inherited. Maternal copy can get inherited with the defected region or the silenced gene or the deleted gene. Here I've given you the example because a few people were confused regarding the maternal inheritance and this Angelman syndrome. So the question was that certain students were saying that in Angelman syndrome it is only manifesting when passed on by the mother. And similar in the maternal inheritance also, if a particular defect is passed on by the mother, then only it get expressed. If passed on by the father, then it won't get expressed. So I'll tell you the difference here very clearly. So first of all, here you can see the father was affected. It has passed to its half of the offsprings, and both the offsprings are carriers. So first of all, the first major difference you will observe between the Angelman syndrome and maternal inheritance is that maternal inheritance is first of all it is the mitochondrial inheritance in which the mothers only passes on this inheritance. So in each case, you can see if mother is affected, mother is carrying that particular trait, then she is passing to all its offsprings. Whereas it is not in the case of Angelman syndrome. Note, it is not passed to all the offsprings by the mothers. So in mothers, you can see here mother is passing. In this case, it has passed to all the offspring, but in this case, it has passed to one of its offsprings. So first condition is that in maternal inheritance, mother will pass this to all its offsprings. So firstly, it is not it is not met in the Angelman syndrome. Second is only mother will pass. Even if father will contain that trait, but still it will not pass. But in this case, you can see in the Angelman syndrome, father was the carrier and it has passed this to two of its offsprings and they are now the carriers. So in this way, maternal inheritance is only passed by the mother at every case, and father cannot pass anything. Even if father passes, there will be no chance that some carrier will be formed. So this is the second difference between the maternal inheritance and Angelman syndrome. So I hope now it is clear that what does it mean. And if we talk about Angelman syndrome, then it deals with the chromosome number 15, which is the autosome and which is present in the nuclear DNA. But maternal inheritance deals with the mitochondrial DNA. So these are the main differences between the Angelman syndrome and maternal inheritance. So you should not get confused between both of them. Okay. So now let's move further. Now let's look the question number 8, which says that mouse models are frequently used to characterize the human gene functions and recreate the disease pathology. While the monogenic disorders are frequently modeled in mice, only a few have attempted to model the polygenic disorders in mice because. So now we know that we use mouse models to, characterize human gene functions. So they are saying that for monogenic genes, for monogenic disorders, we have used them, but we cannot use them for polygenic disorders. So what do you think? What could be the possible reason behind this? So option one says, not all the risk alleles contributing to polygenic disorders are known in humans. The risk alleles could be in the known coding regions, and such regions may not be conserved in mice. Option C says mice lines are used for such studies are genetically homogeneous, isogenic, and hence they cannot be used to identify the multiple alleles contributing to a disease pathology. And option 4 says all of these. So what do you think? Why we can't use mice models for polygenic disorders? Okay, few students have answered option 4, all of these. What about others? Okay, let's see. So yeah, option 4 is the correct. That is all of these, because not all the risk alleles contributing to polygenic disorders are known in humans. So first of all, in the Human Genome Project, we have sequenced all the genes, but till now we don't have complete information of function of all the genes. We don't know. For certain genes, we don't know the functions. And when we will look for the omics, then I'll tell you that means how complicated is the genomics, transcriptomics, proteomics is. So I'll tell you that why it is difficult to characterize the functionality of genes. So first of all, risk alleles contributing to polygenic disorders are not known in the humans. Then comes the risk alleles could be in the known coding regions, and such regions may not be conserved in mice. So when we talk about this model organisms, so why we use model organisms? Because they represent some kind of similarity. But it doesn't mean that the mouse will contain the exact sequence as the humans. So there could be variations in the known coding regions. So even if um, risk alleles are present in the known coding regions, then as they are not conserved in mice, so you won't be able to detect it. Then mice lines are used for such studies as genetically homogeneous or isogenic. So hence they cannot be used to identify the multiple alleles contributing to the disease pathology. So this is also the reason that why we can't use the mice models to model the polygenic disorders. So I hope it is clear that why the correct answer is all of these. Okay, so now let's move further. So now question 9 says, which one of the following statements lists the goal of HapMap project? So I've told you earlier that what is HapMap project, why it arrived, why we used it. So option 1 says to determine the common patterns of DNA sequence variation in the human genome. Option 2 says to identify the common variants likely important in the risk of the common disease. Option C says to make the genome related information freely available in the public domain. And option 4 says all of these. So what do you think? What are the common goals of this HapMap project? Okay, so now let's see. So the correct answer is all of these. So by using this HapMap project, we can determine the common patterns of DNA sequence variations, because we know that what are the haplotypes. These are nothing but these are the a combined set of SNPs. So SNPs are nothing but these are the single nucleotide polymorphisms, or you can say single nucleotide variations that leads to polymorphism. So in this way, we can determine the common patterns of this variation. Then to identify the common variants likely important in the risk of common disease. So yeah, in different disease also we can see that which variant is involved. So we can use the HapMap to check for the variant that could be associated with the disease. Then to make genome related information freely available in the public domain. So as we know that Human Genome Project provides you information of the entire genome, and that is freely available. Now to to isolate the SNPs and then analyze it, so it is bit complicated. So in this, what they does is they have analyzed it themselves and they are providing with the information of variations. And we also know that not just SNPs but the tagged SNPs that are very much specific that could represent the SNP itself. So in this way, if it is available at the public domain, you can simply use it. You don't have to repeat all the analysis. So it has made this work accessible and freely available. So that's why these are the goals of the HapMap project, which it has achieved. And it was launched in October 2002 as a scientific effort to identify the common genetic variations among people. Now let's move to the next question. So the question says, which one of the following statements distinguishes a monogenic disorder from a polygenic disorder? So option A says DNA sequence variation contributes to the disease onset. Option B says can affect either sex. Option C says the affected individual can transmit the disease causing allele to the next generation. And option 4 says none of these. So what do you think? Which one of the statement helps to distinguish between the monogenic disorder from the polygenic disorder? We have already have idea about what are the monogenic and polygenic disorders. As the name suggests, monogenic means If a particular disorder is resulting as a means arising as a result of a single gene, is caused by the single gene modification or single gene variation, or as a result of single gene. And if we say polygenic, that means multiple genes are involved in a particular disorder. Okay, few students have mentioned option 4. Now let's see what is the correct answer. So yeah, option 4 is correct. That is none of these. Because if you see the first three options, so they are very common. That is observed both in monogenic and polygenic disorders. Because in both of them, DNA sequence variation is the one that contribute to disease onset, and it can affect either sex. And option 3, it's saying that the affected individuals can transmit the disease causing allele to the next generation. So yeah, in both of them, the individuals can transmit. So we cannot use any of the statement to distinguish. So in this way, correct answer is none of these. So if we say monogenic disorder, then variation in one particular gene only, and examples are sickle cell anemia and cystic fibrosis, whereas in polygenic disorder, variance in the multiple genes at the same time, and examples are obesity, diabetes, and heart diseases. Now let's look to the question number 11. So it says that in a typical case control study, for gene association, the subjects in the case group are expected to differ from the control group in. So when we are looking for the case and control studies for the gene associations, so what do you think? What should be the difference between the case and control group? First is age, two is ethnicity, C is genetic makeup, and D is lifestyle. So means when we say case and control, then we try to compare them. So control is the one which we have already about, then which for which we have the information, and cases which we study. For example, if we want to study the disease, then in that case control will be the individual who is healthy, and case will be the individual who is unaffected, sorry, who is affected, who is diseased. So we will compare the control and case to identify the defective variation that is causing the disease, or to see the gene that is involved in the disease, what is the variation between both of them. So in this they are studying the gene association. So what do you think, what could be the right parameter, or what could be the thing that will differ among both? Okay, few students have mentioned option 3. What about the rest? Okay, so let's see. So option C, or you can say option 3, is the correct option, that says genetic makeup. So yeah, because you are studying gene association, so you don't have to deal with the age don't have to play any role in this, because you are studying gene association. So ethnicity is also no role to play, lifestyle has also not any role to play. So genetic makeup is the right answer, and the study design is to identify the difference in the genetic makeup, and hence the rest of the other parameter mentioned are matched to the control group. So rest other things will be same in both case and control, they don't have to vary. Now let's move to the next question, which says, in the positional cloning approach, the co-segregation of a microsatellite marker with the disease phenotype in an affected family indicates. So we have already discussed positional cloning and functional cloning. So what do you think? The option 1 says that the microsatellite marker is involved in the disease phenotype. Option 2 says microsatellite marker is located very close to the gene involved in the disease. Option 3 says that the microsatellite repeat is expanding in every generation. And option 4 says that the microsatellite marker is an unreliable marker for positional cloning. So what do you think if microsatellite marker is co-segregating with the disease phenotype? Okay, so few people have mentioned option 2. Now let's see. So option 2 is the right answer. So when this happens, co-segregation means if two markers are linked, and they are getting inherited together, or they are involved, they have the involvement together. So this means that the microsatellite markers is located very close to the gene involved in the disease, because if they are very close, then only they will be co-segregated, or they can be segregated together. So in this way, option 2 is the right answer. Now let's move to the next question. So in the HapMap project, the term linkage disequilibrium refers to: first option is known random association of alleles at different loci, random association of alleles at different loci, linkage of alleles with a given loci, or known linkage of alleles with a given loci. So what do you think linkage disequilibrium, what does that means? Okay, few people have mentioned option 1. Okay, now let's see. So option 1 is the correct answer, because the linkage disequilibrium, it indicates an allele that has recently increased to high frequency under the strong selection pressure. So in this, now I'll clear you the difference between two terms, that is linkage equilibrium and linkage disequilibrium, and what does they mean. So now let's see. When we say linkage disequilibrium, it means that the loci are statistically associated with each other more than you would expect by chance. So in this you can see there is linkage disequilibrium, that means one allele is capital A, one allele is capital B. So here you see one is small a, small b. So in this, capital A is always observed with capital B. It is never observed with small b. So which means that these two alleles are linked. These two alleles are always observed together. And so that means these loci aren't evolving independently as they have been assuming so far. So these are not evolving independently. These are always seen together. So this is linkage disequilibrium. The probability of observing AB is 0.5, and similarly small b is 0.5. So in that way you can predict, if capital A is present, you can predict what could be the next allele. That would be always capital B. So which means there is known random assortment. It is not randomly assorting. Random assortment would be if capital A could be assorted with small b as well, and small a could be assorted with capital B as well. So in that case, linkage disequilibrium is the known random assortment of alleles at two loci. Then what it is saying, that B is more likely to be associated with capital B is more likely to be associated with small a if we say if they are associated known randomly. When they are associating known randomly, uh, then capital A and small b is more likely to be associated with the capital A than small a. So means overall you can say that it is the known random assortment of alleles at two loci, and the closer the markers, the stronger the linkage disequilibrium, since recombination will have occurred at low rate. So means if more closely AB will be present, then there are higher chances of their linkage disequilibrium. And linkage disequilibrium can only be broken by the recombination or the crossing over. So recombination can only take place if two alleles are farther from one another, if there is some minimum distance between both of them. If they are very close, then it's very difficult for those two genes to recombine. So that's why they will be in the linkage disequilibrium. So now I hope it is clear the difference between the linkage disequilibrium and linkage equilibrium. So in linkage disequilibrium, the genes are linked, whereas in linkage equilibrium, there is equal probability of two alleles to combine randomly with one another. So in this you see that capital A has also associated with the small b as well, and capital B, whereas small a is also associated with capital B and small b. So there is some kind of equilibrium. You can see there is equal probability to observe all the combination of alleles. Whereas if there is no equal probability, you can see only the two allele combination are observed. So in this way, it is a linkage disequilibrium. There is no equilibrium observed. Now let's move further. So this question says the term haplotype refers to: option A says a group of individuals testing positive for a specific antibody, a molecular biology method used to track the inheritance of genes in family, a set of markers that are physically linked and segregated together, markers representing haplotype genome. So what is haplotype? We have already discussed this. Okay, few people have mentioned option C. Now let's see. So yeah, option 3 is correct. Haplotype is the set of markers that are physically linked and segregated together. Now next question is, while advances in genomic methods have helped us identify the genes mutated in genetic disorders, the challenge has been to dissect the mechanism by which the affected gene causes the disease. Which one of the following options offers an explanation for this challenge? So the options are: the physiological function of most of the genes are unknown, most of the human genes remain uncharacterized, there are not enough biochemical assays to evaluate the function of all the genes, and all of these. So what do you think, that what is the challenge that we face while we check for the genetic disorders and we identify the genes that are mutated? It is a bit similar as we have talked about the mouse models for the polygenic disorders. Okay, few people have mentioned option D. Okay, this is very easy. I think maximum of you should answer. Okay, so the correct answer is all of these. Because we know that we don't have the complete information of all the functions of all the genes, and there are many genes which are still uncharacterized, and we don't have biochemical assays to evaluate some functions. So yeah, so option D is the correct answer to this question. Now let's move to the next question. So this says two distinct genes that are likely to be inherited together are called. So you must remember they are saying two distinct genes. Option A is alleles, option B is polygenes, option C is linked genes, and option D is none of these. So what do you think, if there are two distinct genes and there is a probability that they both will be inherited together? So what does that mean? What does those two genes called? It's very easy. Few students have mentioned option 3 in the chat, and few students are confused with option 1. Okay, let's see. So the correct answer is option C, that is linked genes. So whenever two distinct genes have higher probability to be inherited together, they are known as linked genes and they are present closer to one another. So if two genes are located on the same chromosome and in close proximity, then they tend to segregate together and do not obey the law of segregation. Hence they are called as linked genes. So why it is not allele? Because allele is the alternate copy of a single gene. When the allele when we say allele, then there should be a single gene. It is the two alternate copy of gene. For example, if there is gene for the height, then the one allele should be for the tall and one should be for the dwarf. So that is the allele, means two alternate copies of a gene. So here they have said two distinct, that means there are two genes. So linked gene is the right answer. I hope it is clear. Let's move to the next question. That will be the last question for this session, which is which one of the following statements is true? All DNA variations are mutations, all mutations affect survival, all variations are in intergenic regions, and some variations may increase fitness. So now what do you think? This has also been discussed in previous sessions. By the time you should have idea about this. Okay. This time many students have mentioned option 4 as the right. Now let's see. Okay, it's right. So the true statement is that some variations may increase fitness. So now let's see. So option A says all DNA variations are mutations. It's not right, because not all DNA variations are regarded as mutations. There could be certain variations, such as if we talk about imprinting, there should be certain variation that arises due to deletions or epigenetic modifications. So there are certain silencing events that happen, or some modifications that occurs at the level of post-transcriptional modifications, or some functional group has attached. So in that case, there is a variation, but it is not regarded as mutation. Now all mutations affect survival. So it is also not the case. There could be certain mutations which offer some benefits to the offspring or to the individual. So this is also wrong. All variations are intergenic regions. This is also wrong, because the variations can also happen at the region where the genes are present, not always at the intergenic regions. So hence the option 4 is the correct, which says some variations may increase fitness. So not all variations, but there could be certain variations that can increase fitness. So the answer says that variations provide the raw material for evolution, which may turn out to be useful for the organism. Because if you see in the perspective of evolution, then you see the ancestors that were present, and now as the humans, you can see if we compare us with our ancestors, we see that there has a lot been variations that has occurred, and not all these variations were harmful for us. It has provided us with different advantages. We are better than our ancestors. So those mutations has only provided us with the ability. That's why we have adapted to newer conditions. We have increased our fitness. So this is all about the MCQs. And now we will be talking about the problems that were there in the session 3 about Prader-Willi Angelman. And at last we will be talking about the omics. And then we will sum up and we will end the session. So if you have any doubts, you can ask in the end. So now if we look at the Prader-Willi and Angelman syndrome, then we can see that as the name suggests, how to remember them. So Prader-Willi, its name starts from P. So you must keep this thing in mind that in Prader-Willi there will be paternal silencing or paternal imprinting. And whereas in Angelman, angel, so mother, so maternal imprinting. So this thing you can remember that in Prader-Willi, the paternal gene will be silenced, and in Angelman, maternal allele will be silenced. So you will see that as in Prader-Willi, it's whenever it passes on from the mother, then the offsprings will always be the carriers. This will never manifest when passed on from the mother. It will always manifest when passed on from the father because it is a paternal silencing or paternal imprinting. So whenever father will pass this, father is the carrier here, father is having the deletion in that region. So all its offspring will show the Prader-Willi syndrome. Whereas if we talk about Angelman, then the scenario will be opposite. That whenever the father will pass, then the offsprings will be the carriers, but whenever the mother will pass, then the offsprings will be affected. So it can either be the 50% offsprings or few of the offsprings, but they will never be the carriers. They will always be affected to whom this deletion will be passed. So, so what explanation they have to make here? They are saying that segregation of the imprinting center deletions. So in this, there is a deletion made in the imprinting centers for the Prader-Willi and Angelman. So unaffected deletion carriers are indicated by a dot, in which there is deletion but they are unaffected. So a deletion of the Prader-Willi syndrome SRO in the Prader-Willi syndrome family is silent when transmitted through the female germline. So whenever female transfers, it is silent. So after transmission through the male germline, the deletion chromosome acquires a maternal methylation imprint in early embryogenesis, leading to Prader-Willi syndrome. So whenever the male transmits it, there is maternal methylation imprint. So due to which it leads to Prader-Willi syndrome. So that's why the father will always leads to the offsprings with the Prader-Willi syndrome. So the distantly related family members may be affected, although their parents are not affected. So in this way, you can see that although if their parents are not affected, but their offsprings are affected. So here you can see that they were unaffected, they were just the carriers, but their offsprings are affected. So they need not to be affected to pass on this disease. So so note that the individual 313 is at 50% risk for a child with Prader-Willi syndrome. So this has also the 50% risk factor that its child will also be the carrier, or sorry, it will never be the carrier, it will be affected because it is the father. Whereas individual 3,3 and 3,8 are not. So if you see 3,3 and 3,8, these are the females. So female will never pass this um, deletion. If they will pass, it will remain silent in their offsprings. So here you can see they are not even the carrier also, they are silent. Okay, so if we look at Angelman syndrome, so what they are saying in contrast, an IC deletion, that is imprinting center deletion of the Angelman syndrome SRO in the AS family is silent when transmitted through the male germline, but prevents the establishment of the maternal imprint when transmitted through the female, leading to Angelman syndrome. So it's saying that when a father will transmit, then it will remain silent, but as soon as the female transmits it, it becomes visible and the offsprings get affected. So I hope it is clear. Whenever you see any pedigree, now you can see clearly that in Prader-Willi, the females will not pass this to their offsprings. The females, whenever they pass it, the offsprings will be having the silent deletions or they will be the carriers. But whenever the male passes this, then offsprings get affected. They will never be the carrier state in between. They will be always be affected. Whereas Angelman syndrome, so in this, the offsprings will manifest this Angelman syndrome when it is passed on by the mother. So whenever you see any pedigree, now I think you can distinguish between both of these pedigrees, between Prader-Willi and Angelman. And I hope now you've got the clarity that what does the imprinting means and how does it influence. So now let's move to the final concept, that is of omics. So when you say omics, omics is nothing but it is the combined study. So when we talk about the central dogma of biology, which we have discussed in our first session, so what is central dogma? That is the flow of information from DNA to proteins and further on. So when the DNA level, till DNA level if we study, it is known as genomics, which involves the genomes, genes, their functions, their structures. Then we move another level, that is transcription, where we have transcribed this DNA and now it has formed mRNA, tRNA, rRNA. So the study at the level of RNA is known as transcriptomics, which involves transcripts. Then comes translation, which converts mRNA into proteins, and these proteins, these translations. So any study that is related to these proteins, it is means that involves the translation, especially when we translate mRNA with the help of tRNA and ribosomes. So that is known as translatomics when we deal with these. But when proteins are formed, then the study that is performed at the level of proteins, that is proteomics. And now these proteins can be categorized into multiple categories. So there are certain proteins which play important role in the enzymatic reactions, and they are the metabolites. So those proteins, when we study the special category of proteins that are involved in the metabolism or enzymatic reactions inside the body, helping the metabolism, so they are known as, that study is known as metabolomics. So these are the fewer omic approaches. So now let's look into little detail. So there is an integrated omics approach in which all of them are integrated to one another. So in genomics, we look for the structural functional analysis of genes and identification of genetic variants. Whereas in transcriptomics, we look at the gene expression, profiling, functional regulations, and differential expression of genes. So now I'll tell you the thing which I was mentioning earlier, differential expression. So what has happened before the human genome was sequenced, before Human Genome Project, people roughly have idea about the proteins that were synthesized. So there were approximately one lakh, you can say. So up to 1 lakh to 2 lakh proteins were there. So the confusion was that how many genes would be involved in these proteins, because we are going to identify the genes. So it was believed that one gene can only code for a one protein. But what happened with passing of time, when human genome was sequenced, then it was identified that there were only 22 to 25,000 genes that were present in the human. So now this question is surprising. It was very surprising, and the question arose that how this limited number of genes, in approximately 25,000, forms the proteins up to 1 lakh, 1.5. So how does this variety, this variation occurs? How these diversity has formed at the level of proteins? So when studies were performed, it was found that there were different processes. One such process was alternative splicing, which was majorly contributing to this diversity. So what happens in alternative splicing? So the eukaryotic genome has this advantage that it contains introns in between it. So the higher eukaryotes shows the splicing, and it was absent in the prokaryotes, but it could be observed in few viruses. So what happened in splicing, as human have exons which are when separated by introns. So whenever we remove the introns by these spliceosomes or by the cell splicing introns, so the exons are joined together. But in certain cases, the exons are not joined in order. Certain exons can get skipped. So in that case, or in certain cases, two exons can be, means introns would not be removed. So intron gets retained between the two exons. So in that case also, so different combination of exons can be formed. That is known as transcripts. So when from a single DNA, if multiple mRNA transcripts could be formed, so that is as a result of alternative splicing. For example, if we have 5 exons, 1, 2, 3, 4, 5, and if there are 3 transcripts, so in each transcript, one transcript would have 1, 2, 3, 5, in another it should be 1, 2, 3, 4, and in another 1, 2, 4, 5. So in this way, you can see there is a different combination of exons that is present. So this is the way how a single gene can form multiple transcripts, and then these transcripts will convert into the proteins, and those proteins will be regarded as the isoforms because they are arriving from the same gene. So this differential expression means that from a single gene, the different transcripts would be formed. They have different expressions in different tissues. So there could be certain transcripts which are specific for brain. There could be certain which are specific for some other region. So when we talked about Angelman syndrome, then we talked about the deletion in the UB3A. So we said that this gene is means if it is coming from the mother, then it get expressed in the brain. But when it comes from the father, it won't get expressed. So when we say gene, so we are talking about its transcript. So it from mother, that particular transcript is forming, and that was not forming from the father. So that is specifically expressed in the brain when coming from the mother. So when that particular gene transcript is missing, so that individual or the offspring suffers from Angelman syndrome. So this is the way that how this diversity has occurred. And there are other factors as well, alternate splice sites and then alternate promoters. So that could also be the possible reason for this diversity. Then comes proteomics that involves protein identification, quantification, and post-translational modifications. Then we move a little level above is the metabolomics, which involves profiling of metabolites, hormones, and signaling molecules. Then comes phenomics, which deals with the phenotypes. So it could be morphological, molecular, physiological, and biochemical characterizations. Then come ionomics, which deals with the study of ions. For example, identification and characterization and distribution of elements. So these are all kind of omics, and this is all about it. Now let's look at the approaches or the techniques that are used to study them. So for genomics, we perform whole genome sequencing, PGM, NGS technology, quantitative trait loci. Then in transcriptomics, the study means the techniques which uses de novo sequencing or next generation sequencing, microarrays, RNA sequencing, RT-PCR. So these methods are used to study the transcriptomics. It involves mRNAs. Then comes lipidomics, which involves study of lipids. So for this we use UPLC, MSMS. MS is nothing but it is mass spectrometry. Then LCMS, liquid chromatography mass spectrometry, and chemometrics. For proteomic study, we use MALDI-TOF. So MALDI-TOF is also a technique which comes under mass spectrometry. It is nothing but it is matrix-assisted laser desorption ionization time of flight. It is a special type of mass spectrometry method. Then comes 2D gel electrophoresis. So these two approaches will be used. Then for metabolomics, we use LCMS based. LCMS liquid chromatography mass spectrometry based. GCMS based, that is gas chromatography mass spectrometry based, and NMR, that is nuclear magnetic resonance. So these are the different techniques which we use. We won't go into the detail of these techniques as this is not the part of your interest in this genomics. So let's move further. But for your idea, I've given you the clarity that these are the techniques which we use to study them. And this is the workflow that how we move from the genotype to phenotype. So genotype means where we look at the genomics and transcriptomics, proteomics, metabolomics. So this is all the information that we have about the genotype. Then we look into the clinical and imaging data sets. We perform fluxomics, ionomics, microbiomics, glycomics. These are all the analysis that are performed on this data. We use the machine learning methods, deep learning methods, or statistical approaches. And at last, all the omics are integrated together and transomics. And then we, for analysis, we can design novel networks. So in the terms of networks, what we does is that these networks could be any networks, either gene regulatory networks or protein-protein interaction networks. So in this, so network is made up of nodes and edges, where nodes represents the entities and edges represents the relationships. So for example, if there is a protein-protein interaction network, so you will place two circles that will be the nodes. So those two circles will be the proteins. And if they will be interacting with one another, then there will be a line joining those two circles that is known as edge. So in that way, you construct a network that which two proteins are interacting and how they're interacting. Then novel pathways, we can also design the pathways. If a particular protein is involved in metabolism, then there is a certain pathway, then we can construct that which genes are involved in which pathway. Then we can model and predictions. We can make certain predictions by comparing it. We can also discover the biomarkers. Biomarkers is nothing but if a particular disease is caused by a certain variation, so that variation would be a biomarker for that particular disease. And then ultimately we can it will help us in the prediction of disease and health, and ultimately we'll get the phenotype, that what will be the expression. So we can relate genotype to phenotype by using these omics, and we can also go from a bottom up approach where we have the phenotype and we can go to the level of genotype, that what would be its genotype. So in this way, this integrated omics workflow helps in the study. So with this, we end your session 4 here. It is all about the week 4 content. And if you still feel certain doubts or any problems, then you can comment in this video in the bottom. I'll try to resolve your doubts, and good luck for your exams. And you can also comment the topics which you want me to clear. So thank you so much. Now I'll end this session.